bring it up.
Okay, I wasn't going to read off the screen, but so today we're meeting on Lutruwita Aboriginal land, sea and waterways, and I acknowledge with deep respect the traditional owners of this land, the Muwinina people. We pay respect to elders past, present and emerging, and to the many Aboriginal people who did not make elder status. Importantly, I think given our focus today and in what we do on food and natural resources, I acknowledge the wisdom of traditional owners in caring for country for thousands of years. Thank you.
So I'd like to invite the first student who will be presenting today. Our first student is an honors student, Luorui Li, and she'll be speaking on natural bioactive compounds for reducing methane emissions in livestock animals. Luorui.
Hello, good morning, everyone. My name is Luorui, and today I'm going to present my honors research project titled Novel Natural Bioactive Compounds for Reducing Methane Emissions in Livestock Animals. So first of all, I want to raise the problem. That is, methane is a major greenhouse gas, as you can see from the statistic. Around 28 to 36 times more potent than carbon dioxide, according to the reference. Methane emissions are from ruminants, are significant contributors to global greenhouse gas emissions. Reducing methane is without compromising animal productivity is a key goal for sustainable livestock systems. Our research content is conventional studies like chemical inhibitors, issues such as cost, safety, and microbial resistance. As a result, there's an increased need in natural bioactive compounds. So today I will be introducing two, but for the intellectual property consideration, the specific bioactive compound name will not be will be referred to lactic acid bacteria and iron binding protein. The lactic acid bacteria is known to influence fermentation profiles by promoting beneficial bacteria that increases the VFA production linked to reduced methane, and the iron binding protein has antibacterial properties that binds iron from the previous study. And for the aim and the research questions, the aim is to investigate the effects of iron binding proteins and lactic acid bacteria on rumen fermentation. I don't know why it's automatically skipping, but yeah, are related to methane reduction. And the three questions that I've reasoned is, firstly, can iron binding protein and lactic acid bacteria influence the rumen fermentation patterns? And what is the optimal concentration of iron binding protein and active lactic bacteria for improving fermentation characteristics? And the third, at what time point would this yield the most significant fermentation outcome? For the method overview and experimental setup, firstly, we collect the rumen fluid from the local abattoir, and then we immediately transfer the samples into the anaerobic condition to the lab. And for the treatments, we set up four. The first is the blank, which is just the rumen fluid itself, and the control, the rumen plus the buffer sample, so it will be stable for the observation, and the lactic acid bacteria and iron binding protein set at 3 concentration respectively. And for the incubation and the sampling intervals, we incubate at anaerobic conditions that suits the rumen fermentation condition. And we collect at 0, 12, 24, and 48 hours, 4 time points to do the samplings. As for the measurements, we did the pH measurement, microbial counts, and VFA for current status. And we have and already have the results, and we have do the DNA extraction and sequencing and gas sampling, waiting for the analysis. But today I will be presenting the results of the first three. As for the pH, as you can see for the both iron binding protein and the lactic acid bacteria, caused a gradual pH decline, which is expected in the active fermentation. Notably, the higher doses in gray color maintain stable pH with a biological range, suggested controlled fermentation rather than excessive acidification. And the second result is the microbial count over time by concentration. The lactic acid bacteria treatment showed the highest microbial count at the 12 hours time point, indicating active growth. And so as the iron binding protein, even though it's not quite significantly different, but you can still observe increase, suggesting that it may support the fermentative microbial growth or suppress methanogens, indirectly benefit other bacteria. And the blank and the control remains slow and steady growth. As for the third result, for the volatile fatty acid, we measured the total one and the separate one as well, but I want to present the most significant rise, which is the acetate and the propionate production. The more, and as you can see, it's increased over time as well as over the concentration. It means more efficient energy utilization and better feed conversion, leading to enhance the growth and the productivity of the livestock. And the higher concentration, and also suggests the utilization of more hydrogen for volatile fatty acid than for the methane production. Not only we look into the specific numbers, but also we look into the ratio of these two fatty acids, because according to the previous studies, the ratio between these two decreases, meaning that the lactic acid bacteria and iron binding protein groups reflects a potential redirection of hydrogen away from the methane productions. As you can see, the original control one was fairly high, and then by some treatments, it decreased over time and over the high concentration. As well as the iron binding protein, similar as the lactic acid bacteria, the acetate and the propionate content increased over time by the concentration. But different than the lactic acid bacteria, the iron binding protein it doesn't not mean the higher, the more fatty acid will be provided. In contrast of the ratio also dropped with the high concentration, as you can see. But it also reflects the shift in hydrogen usage towards propionate and acetate, and rather than methane. And the most notable decrease, as I explained, is not the highest, it's the median. So here comes to the conclusion. remember the questions that I froze before. For the first one, yes, both lactic acid bacteria and iron binding protein did significantly influence rumen fermentation parameters in vitro. And the second question is, yes, the most effective concentration were not always the highest, particularly for the iron binding protein, where the median levels produce the most consistent changes. As for the last question, what time points yield the most is in most samples, whether the pH and the microbial counts and the VFA, the 48-hour time point yield the most pronounced effects on microbial growth and VFA production. Thus, so the conclusion can be made that the likely methane mitigation can be made through the treatment, and it can also support productivity as well. And it can suggest anti-methanogenic potential. And for the future research directions, of course, we're going to understand any potential synergistic effects between the iron binding and protein lactic acid bacteria, and also understand the potential mechanisms of action of both products. And in the future, we hope to conduct an in vivo trials to validate in vitro findings. And that's it for my, this is my scientific references, and that's it and this is my acknowledgement. Thank you everyone who supported me for the past year, especially my supervisors and all the people helped in the lab and yeah, thank you everyone who helped along the way. That's my presentation.
So our second speaker for today is John. John is the second honor student, and John will be speaking on the influence of yeast selection on Australian craft single malt whiskey, new make spirit character.
So my devious plan to have a recording play has obviously failed. But yeah, my name's John. Today I'm going to talk to you about the influence of yeast selection on Australian craft single malt whiskey, new make spirit character. So in Australia, single malt whiskey is not actually separately defined as a category, but consensus is that it's produced at a single distillery from exclusively malt barley, and it is a legislative requirement for whiskey that it be aged in a wooden cask for at least 2 years. Separating craft whiskey is a bit nebulous, but broadly speaking, craft distilleries produce about 1 to 2% of the output of a commercial distillery, meaning that quality is an economic necessity. That was convenient timing. Regardless of scale, single malt whiskey is produced through a set of common steps. First, barley is malted to induce the production of amylytic enzymes. Next, it is brewed through sequential addition of hot water, which saccharifies starch and extracts fermentable sugars. Then it's fermented through the addition of yeast. The alcoholic wash is then distilled, usually twice, which is then matured to produce whiskey. The stage of the process at which the largest number of flavour active compounds, including esters and other volatiles, are produced is fermentation, most of which are the byproducts of yeast metabolism. We'll come back. Craft fermentations usually run longer than commercial fermentations, giving yeast more opportunity to influence the character of the resulting new make spirit, which means that yeast selection represents a lever with high potential for character manipulation for distillers. This has been explored a little bit in the literature. Broadly speaking, brewers' yeasts have shown the most potential, which include ale and lager strains. Comparing brewers' and distillers' yeast, brewers' yeast tends to ferment with lower attenuation and less consistency, but you do get higher ester output and generally higher levels of hedonic assessment in sensory panels. The literature has suggested, but not explored, the use of co-fermentations, which is a notable dearth in existing literature, and it shows the potential for a best of both worlds potential outcome. where you get high attenuation and consistent fermentation of a distiller's yeast, but with more diverse ester output and higher hedonic acceptance. Come back for me, please. So the aims of the present study are to monitor new make spirit fermentations and determine the key flavour drivers, to compare laboratory and craft scale fermentations, to examine the effects of combining yeast varieties on new make spirit character, and to compare instrumental chemical analyses with human sensory analysis. We won't be looking at the comparison of scale fermentations today for the sake of brevity. So the research questions we're addressing are: how does yeast selection affect the sensory attributes of whisky new make spirit as measured by sensory and qualitative analyses? Does co-fermentation with brewers and distillers yeast outperform yeast substitution for new make spirit character and attenuation? And can instrumental chemical techniques such as E-tongue or SPME GCMS accurately and precisely detect differences in new make spirit character when compared to human sensory analysis? Oh, she went for me automatically. So how are we actually going to assess this? So first, we produced 48 individual brews. All of them went for 6.5 days. Paired fermentations were then distilled to produce 24 batches of new make spirit. The first 12 of which formed batch 1, which looked at different combinations of brewers and distillers yeast, such that we had 4 batches of, so that we had 3 individual batches of brewers yeast, distillers yeast, brewers and distillers yeast pitched simultaneously, and brewers then distillers yeast pitched sequentially. There should be some photos of early fermentation and mashing there, but they obviously haven't loaded. Fermentation metrics were collected every 24 hours. These included pH and temperature, yeast counts, and density. Samples taken from 0, 72, and 144 hours were measured by SPME, GCMS, and E-tongue. We also analysed new make spirit samples the same way. Human sensory analysis was conducted via mapping, which is a rapid sensory method that uses clusters that are self-determined by the participants. Come back. And we also did hedonic ratings at the same time. So apart from a lot of alcohol, what did we actually find? So specific gravity varied quite significantly between different conditions at all time points. Brewers and distillers yeast as a combination and distillers yeast on its own, come back please, showed much more rapid fermentation with a higher level of final attenuation, whereas brewers then distillers yeast as a combination showed the lowest fermentation kinetics and the lowest final attenuation. Looking at the rate of change, between 0 and 72 hours, there was broad significance between all groups. pH didn't show any significant variance at any time point. There was a lot of variance within groups. However, between 24 and 48 hours and 48 and 72 hours, come back please, there was significance found between the brewers and distillers categories, showing potential differences in how they acidify the wash. This was also seen in viable yeast counts, where brewers then distillers yeast and brewers yeast displayed notable differences from the distillers and distillers categories, particularly early in fermentation. Looking at the E-tongue results for fermentation samples, we can see that between 0 and 72 hours for all conditions, there are quite noticeable changes, particularly in umami and aftertaste characteristics. However, the amount of transition converged quite a lot between the 72 and 144 hour mark, indicating that most fermentation changes occur early on. So here we have heat maps that compare pairwise time points for the different conditions. From these, we can see quite a lot of, come back please, quite a lot of significant interaction for umami and richness, which are tied to fatty acid metabolism. Multiple linear regressions showed a really high relationship with aftertaste A and umami, with the change in specific gravity being the strongest predictor. Looking at new make spirit samples and comparing that to fermentation metrics, we see that there's a lower amount of predictability. For GCMS, we only identified 4 compounds that varied significantly, and all of them were pairwise comparisons with brewers then distillers yeast. Ethyl octanoate, come back please. Ethyl octanoate wasn't present in brewers then distillers as a group, which is likely due to variations in fatty acid metabolism. Ethyl benzoate was lower in the brewers and distillers condition, which is likely a result of cleaner fermentations and lower stress. Ethyl 9-decanoate was slightly elevated in the brewers and distillers condition, which again likely relates to yeast stress, whereas ethyl decanoate was a bit more consistent, and it's a usual product of yeast metabolism. Interestingly, there was a lot of variation in naphthalene, which likely originated from the peat as opposed to the yeast that was used in brewing. Its variation implies that there is some degree of matrix effect going on with the expression of different esters. Looking at fermentation metrics as predictors for outputs, we can see really high predictability for ethyl 9-decanoate and ethyl benzoate, as well as ethyl octanoate. Again, the change in specific gravity was the highest predictor. So we produced PCAs of E-tongue and GCMS for the new make spirit samples and put an overlay of fermentation influence. Come back, please. As we can see from the chart, brewers then distillers and brewers yeast were much more influenced by fermentation metrics, whereas distillers and brewers and distillers yeast were less influenced, possibly indicating more subtle influences from larger shifts occurring in a number of compounds instead of 1 or 2. Significant shifts occurring, indicating that the influence of yeast might be more nuanced than larger scale changes. Come back, please. Go on. So analysing the napping data, we found 3 distinct clusters formed. Cluster 0 was driven by mouthfeel, including, come back, please, including saltiness. Cluster 1 was a bit less defined, whilst cluster 2 had higher levels of ethyl 9-benzoate and ethyl benzoate and ethyl 9-decanoate, as well as higher levels of bitterness. Aligning this with a PCA that looked at just GCMS and doing a cluster analysis on both of them, we found 5 individual batches. Come back, please. 5 individual batches that shared a cluster between the 2. This group was defined by lower levels of ethyl benzoate, a higher amount of drop in specific gravity, particularly early in fermentation, and an overall higher hedonic rating, although this difference was not significant. And it didn't align with the groupings of any specific condition. So in review, we found distinct differences in the different categories of yeast used. Brewers yeast fermented with more rapid acidification and a higher diversity of flavour. Distillers yeast had a cleaner fermentation with an overall lower ester yield. Brewers and distillers yeast used simultaneously exhibited a more rapid fermentation and hybridised chemistry between the brewers and distillers categories with relatively diverse ester formation. And brewers then distillers yeast exhibited divergent chemistry with much slower fermentation but much more pronounced ester expression. Looking at the human sensory data, we found partial alignment between napping and GCMS data, although human sensory perception is difficult. Come back, please. Is difficult to assess directly. Some of the nuances are yet to be explored in the data. Combining E-tongue data into our grouping matrix may help us to further unpack the data, though. All right, now you can transition. Thank you. So I just want to acknowledge both my supervisors, Naomi Vinden and Samantha Sawyer, have gone above and beyond to enable me to conduct this research from Somerset while enrolled in Launceston. It's been greatly appreciated. Also, all of the other TIA staff have been more than happy to add their input, helping us with things like GCMS methods or understanding some complicated stats. My family for putting up with me for the last week in particular. And the distillery dog for reminding me that even at the end of a 20-hour day, nothing in life is more important than playing fetch.
So we're now switching over to the Master of Ag and Food Science students for their presentation. And the first on my list here this morning is Pramila. Pramila will be presenting on feeding behavior of lactating dairy cows, grazing pasture in different farmlets. Welcome. Yeah. And make sure you keep your eye with so that you can take note of your time.
Okay. Good morning, everyone. I am Pramila Bhandari. Today, I'm going to present on research in progress, feeding behavior of lactating dairy cows, grazing pasture in different farmlets. This is the outline for my presentation. Let's start with the introduction. Usually, milk production rely on high quality pasture intake by the cow and how they graze on the paddock. And grazing efficiency is influenced by three major factors, that is cow breed, lactating stage, and most importantly, pasture quality and quantity. And usually cows spend half of their life on the grazing or ruminating. And the key intake factors are grazing time, bite rate, and bite mass. And cows spend up to 90 to 95% of their time on three different activities like grazing, ruminating, and resting, and the processes of recycling. And most of the this grazing and ruminating was highly influenced by diet type, weather, daylight, pasture quality, and physiological state. And ruminating will be peak after 4 hours of feeding while they are taking rest. And we can see there is differences between two different breed, that Jersey cow, which Jersey cow that is higher bite rate and mastication efficiency, and Holstein Friesian cow, which is very large in size and they need high energy and rely more on supplements. The major nutrition source for the cow is pasture, and that should be in higher quality. That play a vital role for their development and other stages. And high quality pastures should be leafy, low steam, and easily digestible. And most of the pastures were boosted by nitrogen fertilizer. And when nitrogen fertilizer work, when the soil temperature will be higher than 8 degrees Celsius, but if the soil temperature will be lower than 4 degrees Celsius, then it will be least effective than 8 degrees Celsius. And in the preference of Tasmania, most of the farmers use two pastures, that is ryegrass and white clover, due to the climatic condition in the Tasmania. And high protein pasture often lack energy. That will reduce the nitrogen use efficiency. That's why WSC, water soluble carbohydrate in the pasture will improvise the protein synthesis, and that will help to increase the milk production. And in precision dairy farming, they use a lot of technology for monitoring cow individual health and productivity. Likewise, variable sensor, like nowadays they use collar ear tag to track the activities, feeding habit, and rumination. And in variable sensor, there is accelerometers that will detect the behavior of the cow, which will be accurate almost 98%. And also there is RFID, radio frequency identification, that will be identify individual cow, how they are grazing and where they are going. And they also help to enable the detect the health issue of the cow, and also it helps to collect the data so that there will be less use of labor in the farm. And my major aim to research this project is to study how different pasture type and nitrogen fertilizer level affect the feeding behavior, like grazing, ruminating, and resting of lactating dairy cow in Tasmania, and how does the relate to milk production, body weight, and body condition score, and improve the pasture-based dairy management strategies. And the hypothesis is pasture composition and nitrogen regime do not significantly influence cow feeding behavior and productivity. This is the layout of my project. We can see here. This one is eight different block with the 4 different farmlet, and we can see here blue one is farmlet 1, farmlet 2, and purple farmlet 3, and green is farmlet 4. And we put different nitrogen dose on different farmlets, and number of cows were also different in different farmlets. And we can see in farmlet 1, 2, and 3 have 29 number of cow, but farmlet 4 have 22 only. And nitrogen dose is in farmlet 1 was quite higher, and farmlet 2 and 3 was half of the farmlet 1, and farmlet 4 there is no use of nitrogen. And this is how land preparation was done, and this is the irrigation. Yeah, and this picture, the first one is rotary dairy with milk flow meters in 50 bale, and this machine's name is Devlalal, and where we can see here how much milk production was done, 13.17 liters, and while they we can see in the picture B, where body weight, scale, and body condition were captured by the camera when they walk into the milking time. And here we can see the ruminating and grazing pattern of the cow. And the first one we can observe here, one bell-like object. This one is halter. So we can monitor how the cow were grazing and where they are grazing. And in this picture we can see this one picture is a mix of ryegrass and clover, which was grown in farmlets 1 and 2. And this one is plantain, clover, and ryegrass, which was grown in farmlets 3. And this one is farmlet 4, where is mix of number of pasture. We can see some plantain, some seeds, some grass, and clover, everything over there. And in this picture, we can see how they are selecting the pasture. They are like skipping some patches of the grasses, and they choose the clover and other ryegrass, and they skipping some grasses over here. And after grazing, they are taking rest for ruminating. Yeah, and we select 4 different time periods for data, for observation of the cow. And usually we prefer after peak lactation period for the observation, because during higher period of lactation, the energy of cow will be lower, and they don't want to feed more vigorously. That's why we don't want to feed them silage during that time. And because we are observing their grazing habit, that's why we choose after the peak lactation period. And we have to ensure that consistent pasture only as a forest condition when they are grazing. And after that, we remove outlier because some cow were sick and some cow are in heat and heat condition. And when they are heat condition, they are more in moving rather than they are grazing and ruminating. And this will help to improve the data accuracy and reliability. Here we can see this one. This picture is from farmlets 1, where they are grazing properly. But after slowly, they are in picture B. We can see this is a farmlets 4, where they are also grazing properly. But in the grazing in farmlets B, picture C and D, we can observe that how they are skipping the grasses, which is lower than the clover and the grasses in the patch form. And after that, in picture D, after finish all this clover and ryegrass, again they are going down for residual to feed. And here we can see this is farmlets 1, where they are grazing uniformly. But in figure 4, in big figure B, we can see some clover patches and grass clumps. But in figure C, we can see cow graze down to the residual, residual than the grasses. They chooses over. They choose clover over the grass. Now jump into this result. And this is the box plot. And we can observe here milk yield in 4 different time. We don't find any significant differences. And we can find any new things among 3 farmers, different farm. Same result we observe in body weight too. We didn't find any differences between farmlets in different time. A body condition score also like that, like same. Here in grazing, result was similar before, but sometimes here in the November and December we observe a slight higher amount of grazing rate in farmlet 2 and 3, but not so much differences. I didn't find any difference between the grazing pattern two in 4 different time period. Resting also like same. And in ruminating also like same. I think it is due to the selection of grazing habit of cow because we have different pasture in different farmlets, but we got the same result in 4 different farmlets due to their maybe due to their grazing habit. That's why we are searching for new procedure how to tackle with this. If there is any difference or we are confused in the result, or we have to get other technology for determination to grazing habit for that. And in moving also like same. This is continuing work. And this project was for 3 different years, 3 different replication, and still there's only 2 years. Still we have 1 more year for more data. And we are thinking that all these are 28 average data. Now we are thinking that there is any difference between 3 days mean if there is any difference between 3 days grazing or other resting or ruminating in data, so that we are thinking to analyze mean of 3 days rather than 28 days. And also we are thinking to analyze the botanical composition between different families or different paddock. And I think overall this thing we will get some more significant data in future. In conclusion, milk yield and body weight was similar across all the treatment, and selective grazing may balance out feed quality difference that may occur due to different level of synthetic nitrogen applied and species mix used in farmlets trial. I would like to thank my supervisor, Peter and Jack, and Dairy Australia, Tasmanian Institute of Agriculture, and TIA. Thank you so much.
All right, thank you very much,
Thank you.
Pramila. I would like to invite Umma, who will be presenting on fungicide resistance in alternaria, solani, and no, it's different from what I have here. Okay, yeah, well, introduce yourself and your topic, and then take it from there.
Sure.
Thank you.
Good morning, everyone. This is Umma Ruman Brishty, and today I'll be presenting my master's research project, which is fungicide resistance in Alternaria solani and sclerotiorium, the role of microconidia in sclerotiorium pathogenicity. So these pathogens are very important for potato production. So if we go for the Alternaria solani, this was first described by Ellis Martin in 1882 as Macrosporium solani, and it is a very dominant pathogen on potato, causing early blight, and it also causes crop loss, fluctuating between 5 to 78%. And if we go to the next one, that is sclerotiorium. So Madame Emily Bird in 1837 first described the fungus, and she named it s.sclerotiorum. And this is also a serious threat on potato production. It causes stem rot or white mold. So it also causes a considerable crop loss that reached up to 60% as reported. If we want to take a look on the disease overview, so for Alternaria solani, it produces its characteristic lesions with concentric rings on the leaves and stems. And it also occasionally occurs in tubers if the tubers come contact to the conidia, which is the asexual spores of this one, by which it reproduces. And the disease that really impacts to reducing the photosynthesis in the plants. And for this sclerotiorium, it appears as water soaked lesions primarily, which later on grow into a white cottony growth of fungus. And it can also cause tuber rot as well sometimes. And one interesting thing about this organism is, besides sexual and asexual spores, it can produce microconidia as well. That is a significant interest of my research this time. So how the pathogens are impacting overall? First of all, they're reducing the yield significantly, and farmers are using too much fungicides to control them. So it is definitely increasing management costs as well. And the use of fungicides, they are developing some sort of fungicidal resistance in these organisms. And last but not the least, the use of fungicide, they're impacting the environment negatively. So before undertaking any research, we should know the knowledge gap. So our knowledge gaps are, we don't know the effect of fungicides on different fungal growth stages. And we also don't know the functions of microconidia of sclerotiorium. There come our research questions. Do the pathogens have fungicide resistance? what is the effect of commonly used fungicides on different fungal growth stages, and what is the role of microconidia and sclerotinia pathogenicity. So our objectives are to find the answers of these questions. We really hope this research will bring out some outcomes, and those are the enhanced productivity, reduced production cost, control of fungicide resistance, and reduction of environmental impact. So if you want to have a quick look on the methods, it's pretty simple. So we just segmented our research project into 3 parts. The first one is identification of the pathogen, which we have done through 2 different stages. The first one was morphological identification under microscope, and then we confirmed the pathogen through DNA extraction, PCR, and gel electrophoresis. The second one is isolation of the pathogens. For this one, we isolated the pathogens from the source, and one part we cultured on the fungicide-treated media, and we designed the experiment as RCBT. And for the third one, we only used sclerotinia for this one, as we wanted to know the function of microconidia. So we inoculated on fresh leaves and stems, and also on bruised leaves and tubers. So in the results and discussion section, we can just mention about the first one, Alternaria solani. So we found this very classical conidia of Alternaria solani under microscope. So we did not go through any other further process because this is enough to say it's Alternaria. And for the second one, sclerotinia, we went through the DNA extraction, PCR, and gel electrophoresis, because this is very similar to another microorganism that is botrytis. So we needed to be confirmed that it's sclerotinia or botrytis. So we used 50 base pair ladder and the right-hand side sequence primer for this procedure. For the fungicide resistance part, we used 2 very commonly used fungicides. Those are mancozeb and azoxystrobin. We use the commercial rate and 15% higher than the commercial rate as treatment. And for this part of the experiment, unfortunately, the thing grew. It was not Alternaria. So we suspect that that thing could have inhibited the germination of Alternaria. And we wish to reculture it from a different source, because we tried from the same sources and we got the same result. So we really wish to have a further research on that thing as well that could have inhibited the germination of conidia. But that thing that has germinated all of the treatments, as you can see, like T4, T3 control and T2 as well, which and it didn't grow for the T3, which was the 15% high rate of mancozeb. And for the sclerotinia sclerotiorium, we cultured it from 2 different sources, mycelium and sclerotia, because it has 2 different fungal growth stages that can infect the plants. So for the first one, mycelium, as we can see that all the treatment grew up into 100% germination. But for the first one, mycelium, they grew sclerotia for all of them. But for the second one, sclerotia, they did not grow for the mancozeb treatments for some reason. So this is how it looked in the PD medium with the sclerotia, and the right-hand side, this one only from the mycelium. So if we look at the growth of germination percentage of Alternaria solani over time, we took the data after 10 days of inoculation. So it showed like the azoxystrobin, it allowed the faster spore germination than mancozeb. And the higher rate of mancozeb, it completely inhibited the germination for that thing. It's not Alternaria solani, I told you previously, but that thing, it grew like this. And if we look at the Sclerotiorium mycelium and Sclerotiorium sclerotia, the all treatments reached up to 100% germination. And the elevated fungicide concentration of mancozeb that initially delayed the germination percentage, but it coped up later. And if we look at the number of sclerotia from the both sources, that is mycelium and sclerotia, so we can see that the sclerotia formed from all the media of mycelium, but in case of sclerotia, it only produced for azoxystrobin. So we can say that mancozeb treatments restricted sclerotia development more effectively, but azoxystrobin treatment supported sclerotia formation. For the last part of our experiment, we inoculated the microconidia of sclerotinia into some detached plant part. That was the first one, A. It was a detached fresh leaf that you can see it has already formed some characteristic symptom, but this is too early to say that is it the same thing or not. So we're still looking into this. And that one was the lower part of the leaf, detached leaf, and the figure C is showing the characteristic water circulation that was in the leafing plant. It is not detached. And for the part, for the figure B, that was inoculated on the upper surface of the leaves, but we didn't find any infection for this one. And for the leafing plant, the upper part of the leaf in plant C, sorry, in figure D, it showed some symptoms, but we're not sure about this one because it doesn't look like the characteristic symptom of the sclerotinia. And for the inoculated tubers, we didn't find any symptom of the infection. So in conclusion, we can say that the high rate of mancozeb completely inhibited the unknown growth. In the first one, we tried to grow alternaria solani. And for the sclerotinia, it has resistance to both mancozeb and azoxystrobin. But in case of mancozeb, I think it can delay the germination. And also the microconidia, they cannot affect the tubers, but it might have the ability to infect leaves. Thank you so much, everyone. And I would also like to thank my supervisors, Dr. Callum Wilson, Dr. Jonathan Namponsa, the TI staff from Newton Research Lab as I work there, and also my dearest husband, Najmul Huda, for supporting me all the way. And these are the references you could look at.
So our next presenter for today, and don't worry, very soon we'll be out for morning tea. But our next presenter for today is Shilpa. And Shilpa will be presenting on factors limiting the seed yield of parent lines of hybrid carrot polytunnels.
Thank you. Good morning, everyone. My name is Shilpa. Today, I'll be presenting on factors limiting the seed yield of the parent lines of hybrid carrot in polytunnels. This project was carried out under the supervision of Dr. Elita and industry supervisors, Dr. Cameron and Amy from Seed Purity. Carrot seed industry is one of the important part of the vegetable seed industry in Australia, and it is one of the major producers and exporters globally. Tasmania alone produces up to 320 tons of hybrid carrot under 700 to 800 hectares of land, and polytunnel production is an emerging opportunity for the industry as the polytunnels offers protected and controlled environment to maintain genetic purity and manage the growth of carrot parent lines. Also, stock seeds from polytunnel supports large-scale carrot seed production. So the carrot seed production is done under two production systems, open pollination and cross pollination. Cross pollination produces the hybrid plants, and this system is based on the two types of parent lines. One is the cytoplasmic male sterile line, which is the female line, and the other is the male fertile lines, which is the male lines that produces the viable pollen. And this is generally planted at 2:1 ratio with 2 rows of female plants with 1 row of male plants. In this tunnel, we can see the male line is on the left side of the tunnel and the female line is on the right side. One of the major challenges we are facing in polytunnel is the pollination issue. We are facing the inadequate pollination, which often limits the seed set. And the other problem we are seeing is the seed yield variability with the inconsistent and low yields in polytunnel. So our project aim was to find out where the problem was so that the further research can be carried out. One of the aim was to evaluate pollination efficiency by monitoring the hoverfly activity, which was used as the pollinator in the polytunnel. And the other aim was to identify other yield-limiting factors like pollen viability, pollen deposition, and seed set. So the experimental design, this research was carried out at South Pacific Seeds Richmond polytunnels, and the trial was carried out from flowering to harvest from early December to late February. And the laboratory analysis was done in Seed Purity Market Lab and University Lab. So we used the two tunnels, tunnel 2 and tunnel 9, namely with different variety, but under the similar environmental condition. And the pollinator used in tunnel was hoverfly, Eristalis tenax, with 15,000 hoverfly released per tunnel. Hoverfly is mostly used in polytunnels these days because of their efficacy in pollination. Also, they do not sting unlike bees, and they are good under the polytunnel condition, cool temperature. So this is the tunnel layout, and this is the tunnel layout, and it was similar for both the tunnels. So there were 6 beds of plants, and the male beds were planted at the either side of the tunnel. This blue line represents the each rows of male plants, and these female lines were situated at the center. There was 4 beds of female plants with 2 rows in each beds, and we randomly assigned 15 transects throughout the tunnel in female lines and 8 transects in the male line for the data collection, and each transect was measured of 5 meters along the bed. We also monitored the environmental condition using the data loggers. So this is the carrot flower structure. This figure shows the carrot plants, it's showing the carrot inflorescence, which is made of the number of umbels. And each umbel is made of the cluster of umblets, and each umblet consists of a number of individual flowers. So the data collection was done at a different level during this trial, and some data were collected at umblet level, and some with individual flower, and some data were collected on whole plant level. Materials and methods, we counted the hoverfly numbers to observe the pollinator activity at different times of the day and throughout the flowering season. And then we collected the pollen from male lines to access the viability and the pollen yield from the both tunnels. We did it at 2 times a day at 9 a.m. and 11 a.m. and also throughout the flowering season. So the next step was to pollinate by hand or hoverfly to see if the pollen deposition was good. And this pollen deposition was calculated by doing the stigma mounts and looking under the microscope. And finally, looking for the seed set and yield in both the tunnels. So result, seed set. We observed the seed set data by two different methods. One is the seed set percentage. We calculated this based on the seed set amount on the individual omelets. And the result we saw was the higher amount of seed set in female beds, which are closer to the male lines. As we can see there, F1 and F4 are closer to the male beds, and there are no male lines in the center of the bed. So the seed set percentage was higher on F1 and F4, and this was similar in both the tunnels. And this difference was significant in tunnel 9 and with a similar trend in tunnel 2, but was not significant. And also observing the seed yield data, it showed the similar trend. And this seed yield was calculated by harvesting 40 plants per the beds. And we calculated the yield data from each bed. And it shows the similar trend of higher amount of yield on the female bed closer to the male lines than on the central beds. And we also got the yield data from seed purity, and tunnel 2 was actually good and got 146% of the yield target, while tunnel 9 didn't meet the target and it was just 18% of the total yield target. To know how the seed yield data changed in both tunnels, we looked at different factors, and one of the factors we looked at is pollinator activity. We observed the overfly numbers in both male and female lines throughout the season at different times of the day. And similar to the seed yield trend, we found that the number of overflies in female beds closer to the male lines, that is F1 and F4, was more compared to the female lines that was in the center. And this difference was significant in both the tunnels. We also observed the pollinator activity, that is overfly count, at different dates throughout the season, and we didn't see any trend of the overflies, but generally the number of overflies was more on male lines compared to the female lines. That might be because of the nectar and pollen production in male lines, while only the nectar production in female lines. But interestingly, in tunnel 9, in the later the season, the number of overflies in male line and female line was not much difference, and this might be because of the lower amount of pollen production in male lines in tunnel 9, which we'll explain later in pollen viability. Also, we measured this data at different times of the day, and we didn't see any particular trend along the time of the day. Although we didn't measure the data at late in the afternoon, which might be different from this data at 9 to 1 p.m. because this was the peak pollinator period. And this is the temperature data throughout the season in both the tunnels. So the next step was to look at the pollen production at both the tunnels. And this figure shows that the pollen production was higher in tunnel 2 compared to tunnel 9, which might explain that result in the overfly numbers in male and female lines in tunnel 9. And we also calculated the viable percentage of pollen if it was an issue, and we find out that percentage viability was good in both the tunnels, so this is not an issue. And this figure above shows the FDA test for the pollen viability, and the fluorescent ones are the viable pollens and the dull ones are non-viable pollens. So the next step is to see if the pollen available is actually transferred to the female flowers, or is the pollination even limited in these tunnels. We calculated this data by collecting the umbels and carrying out the pollen deposition test. We mounted the stigmas in basic fusel gel and looked under the microscope. And we found out that by doing hand pollination, we were able to deposit more pollen than by control one, that is hoverfly mediated pollination, and it was similar in both the tunnels. And we compared this result with the seed set result, and this seed set was carried out by we tagged a few of the omelets earlier in the season, and we harvested that later in the season and calculated the seed set percentage. And we found out that the control and hand pollination did not show any much difference in seed set percentage in tunnel 2, which suggests that the pollination by hoverfly was actually efficient in tunnel 2. But it was different in tunnel 9, and hand pollination was significantly producing higher seed set percentage. And this might be because of the lower pollen production in tunnel 9, and the hoverflies were not transporting enough pollen for good seed production. So this is the summary of the results. The first one, pollinator activity, hoverfly number per tunnel if it's an issue. This was not issue as there was enough pollinator in both the tunnels. And we also looked at the hoverfly activity by different types of the day and flowering period. And it didn't show any difference throughout the flowering period. So it might not be the yield limiting factor. But we see the clear difference in hoverfly activity across tunnel beds. So this might be the reason behind yield limitation in polytunnel. And the next one is pollen production. Total amount of pollen available is definitely a limiting factor in tunnel 9, as there was a low pollen production in tunnel 9, but this was not an issue on tunnel 2. Likewise, the viable percentage of pollen was not an issue in both the tunnels, as the percentage viability was quite good in both the tunnels. And observing the pollen production by time of the day, that is on 9 a.m. and 11 a.m., there was not much quite difference in this data, so this might not be the limiting factor. And pollen deposition was an issue on tunnel 9, as we see the seed set percentage data on tunnel 9 was increased by hand pollination compared to hoverfly, but this was not different in tunnel 2, so this might not be the issue in tunnel 2. And we also looked at flowering synchronization, if the male and female flowers are like flowering at the proper time. So it was good, and it is not definitely not a limiting factor. And seed aversion during seed maturation. We calculated this by harvesting some seeds earlier to the harvest and some at full maturity to find out the differences in these two, and we didn't find any significant difference. So there was no any seed aversion issues after the pollination was done. And seed yield across the tunnel was an issue in tunnel 9, as high number was observed in beds closer to the male lines, but not in issue in tunnel 2. So the conclusion we can draw is seed yield was higher in female bed located to male lines, and hoverfly number was adequate for pollination. And pollen availability was an issue in tunnel 9, but not in tunnel 2. And hoverflies demonstrated strong potential as effective pollinator, as evident in tunnel 2, where pollination was nearly at peak level. So the next steps can be exploring positioning male lines in central rows to improve pollinator access and pollen deposition, investigating the performance of pollen output of male lines used in tunnel 9, and using management strategies to ensure adequate hoverfly vegetation in female lines, for example, maintaining good nectar production. Acknowledgements. I would like to thank my supervisor, Dr. Elita, for her support and guidance throughout the research period. And special thanks to Dr. Cam and Amy and the whole seed purity team for their support and technical guidance throughout the research period. Also thanks to Craig Garland and the South Pacific seed team for providing the research site and on-ground support. And thank you everyone for being here today.
All right, thank you very much, Shilpa. Just one more to go, and if all things go well, we should be on morning tea by 11:30. So our next speaker is Michael. And Michael will be presenting on the application of cherry vinegar as natural preservatives and sensory enhancers. You can use the
Right, good morning, everyone. Okay, so yeah, what better topics to preface the tea break after this? So we're talking about foods. So I'm Michael Liong, and just gonna share my findings about my research. So on application of cherry vinegar as natural preservatives and sensory enhancer. And this has been the part of fermented fruits as a preservatives project. And okay, so moving on. So why do we do this in the first place? So as you can see, modern lifestyles force the consumers to find more convenience food. In Australia alone, the convenient food market, including ready to eat, shelf stable, and all that, valued at $3 billion in last year and expected to grow until $4 billion in the next 10 years. And when you see number goes up, brain goes woo. But on the other hand, we have consumers being aware of the side effects of synthetic preservatives, and they are actively looking for natural preservatives like our consumer Drake's here. And okay. Also, in Tasmania, we have lots of cherries, and it's known worldwide. Tasmania has been the biggest cherry producer and exporter for the past, I don't know when. But also, we have lots of second-grade fruits, or like almost sometimes maybe up to 50% of these fruits are unable to be sold. And therefore, one way to combat these second-grade fruits is to ferment these fruits into vinegars. And cherry vinegars have been reported to be rich in polyphenols and other bioactive compounds such as all the organic acids and polyphenols. So these have the antimicrobial and anti-lip oxidation acts as antioxidants, which can be used to preserve food shelf life. Yeah, so along with sous vide cooking, which is a method of cooking with vacuum products and cook in low temp water bath. And this project is done with the Association of Wild Mother Tasmania, which has kindly provided us with their cherry vinegars. And the study aims to investigate the effectiveness of cherry vinegar as the natural preservatives and sensory enhancer in sous vide beef. So first, our research questions was to see how effective the cherry vinegar to inhibit microbial growth and how effective it is to inhibit lipid oxidations. Also, how stable it is and what concentrations needed, and also what impact does it have on the food itself. Right. And how do we do this? Right, so ground meat. So we chose ground meat as our samples. And so plus the treatments, these are the treatments we have. So we start with control, we just add 10% of water. And then we have the 3% plus 7% water and 5% and 7% of cherry vinegars added into it and mixed together. So, and then we put 20 grams of them in a vacuum bag and press into patties. And then we proceed to vacuum pack and cook it in sous vide at 90 degrees for 20 minutes. And we store them at 4 degrees Celsius or refrigerations for 3 weeks. And we analyze them every week. And these are the analysis that we do. So starts pH, drip loss, color, all the antioxidant properties, and then the lipid oxidations, fiable count, and then the volatile assay as the sensory assay. So these are our results and discussions. On the top right, that's the products on after first day of cooking, and then that's the product after 3 weeks of storage. As you can see here, there's a slight difference between the control C and then the 3, 5, and 7. However, so this the color data. So we don't find any significant difference on the samples over time. So we aggregate all the data into just treatments. So that's the L, A, and B values. L representing the lightness, lighter color, the higher the number, the lighter the colors, and then A representing the redness and B the yellowness. So we can see the cherry vinegar treatments does have significant effects on the L, A, and B. So the sample shows lighter colors, lower redness, and lower yellowness. Yep. So we theorize that the lower pH of the cherry vinegars denatures the myoglobin faster, and therefore lower the redness and gives it higher lighter color as well. And moving on to next one. So this is the pH data. The cherry vinegar itself has a pH of 3.54, and most samples doesn't really change or behave significantly over time but the control, I think that's because of the deviations it has. And adding more cherry vinegar does in fact lowers the pH. So we get a control at 5.9 pH, and then all the way to 4.9 or 4.7 with the cherry vinegar 7%. And also in our experiment, we found that we have a slight upward trend happening across the 3 weeks of storage. However, it is not significant. Right, so and then with the pH, we also have this drip loss effect. We found that the cherry vinegar treatments actually increase the drip loss. But however, keep in mind, we're only adding acids in the meat. So I guess if we add binders, this will not be as big of a problem. And 3% does have a slightly lower average, but as you can see on the right hand graph there, the lower pH does decrease the water holding capacity. Right, then we have the antioxidant properties here. So while adding cherry vinegar does gives higher average, but the data shows it fails to show any significant difference across treatment and over time. However, if I were to aggregate the data into aggregate the data across time periods into just treatments, we do have significant difference on 7% cherry vinegar treatment. And while at yes, so Haraf, the literature from Haraf also reported similar results. Adding polyphenols to meat does not always mean to increase the polyphenol content. So up to this point, adding cherry vinegar doesn't seem to be good enough. And then we have the DPPH. DPPH data. Something's wrong with the slides, but DPPH first. Also show the same pattern as the TPC. It failed to show any significant data. And the FRAP So the FRAP does cherry vinegar treatments does increase the FRAP. However, it deteriorates significantly after 3 weeks of storage. Now, but cherry vinegar treatments does significant impact on lipid oxidation, which is what we are looking for. And cherry vinegar treatments does decrease the lipid oxidation since day one, and it's able to keep the lipid oxidation low up to 3 weeks after. Right. And what does that mean for the sensory? And this is the data that we found from GCMS. 100 plus compounds were detected, but these few are selected because they are very consistent across all samples and across all time period. So we see that, so these samples are like most of them are aldehydes, and we have some alcohols and furan. And these aldehydes gives this grassy, and some of them give garlic and meaty flavors. And if on the data, we found that the cherry vinegar treatments have significant effect on the aldehydes produced because the aldehydes produced from the lipid oxidations, and overall control have significant higher aldehyde content and resulted in stronger beefy aroma. And this happens to all the aldehydes, has the similar patterns of inhibition by cherry vinegars. And also in cherry vinegar samples, we have this acetoin compounds, which is not found in any control samples. And acetoin gives this creamy, buttery, yogurt-like characteristics. So in my experience, smelling the product, smells better. And last but not least, we have this microbial analysis. There were no growth found in any samples in the 3 weeks storage of chilled storage, but sous vide cooking seems to be very effective in reducing the microbial load. But the samples kept at room temperature shows growth on control and up to 1,000 times dilutions. And while the cherry vinegar treatments doesn't show any treatments even with lower dilution as well. And then we came back to our research question. So what do we find? Cherry vinegar seems to be able to inhibit microbial growth. It is able to reduce lipid oxidations, and it is pretty stable. So as you can see, that there is no significant difference for TPC and DPPH over 3 weeks. But the reducing power does significantly reduce after 3 weeks. And what's the best balance? I would say it's 5% for the significantly inhibit lipid oxidations and microbial growth. And CV treatments can indeed prolong shelf life and improve the sensory characteristics. And yeah, special thank you for all my supervisors, which without them, I will not be able to do any of this. And to Zubayat, my lab mate, and Dr. Tim Jones from Wild Mother Tasmania. And thank you.
Michael, can we appreciate Michael?
Welcome back, everyone. Thank you for joining us. I'd like to introduce you to one of our Masters of Ag and Food Science students, Thuy Duong Nguyen, sorry.
Yes.
She's going to talk to you about characterizing licking behavior and performance of sheep at a Bromoform, I can't even pronounce it. Bromoform-containing block supplement. Thank you.
Yes.
Take it away.
Yeah, thank you. So good morning, everyone.
So first of all, I just want to say thank you with all the professor and everyone who attend the seminar today. I think you chose the, yeah. Yeah. Yeah, that's good. Yeah. So first of all, I just want to say thank you with all the professor and everyone who attend the seminar today. So my name is Thuy Duong, and today I will present the research project for characterizing intake, leaking behavior, and performance of seed provider seed feed containing block supplement. And my supervisor team is Dr. Ape, Dr. Richard, and Mr. Rohan. And also we have support team from Anadel Farm and Seed Feed Company. Yeah, first of all, let me introduction something about story why we conduct to do the project. So methane from ruminant animal is a potent greenhouse gas contributing to climate change. Methane emissions from grazing system in Tasmania and in Australia as well is a significant concern now, because in Australia, 71% of agriculture greenhouse gas emission are indirect methane, mostly produced in grazing sheep and cattle. And secondly, several feed activities have been identified to mitigate methane emission, including asparagopsis is a red seaweed. And asparagopsis contains the active ingredient bromoform, which is effectively, but we face is a delivery mechanism challenge in grazing system, because in extensive grazing system, livestock have the right to move freely on large pasture, ensuring the dosage of bromoform for animal is so difficult. And in addition, the extensive grazing system, especially in livestock, have a food supply chain that seasonally. So if the animal receive too much bromoform, they will lead to poisoning. And moreover, bromoform is a volatile compound and it's difficult to maintain outside the environment. So if we let it for too long outside, it can be reduced quality or be lost. So that's for the reason we just have a pollution like to prevent the challenge, provide more form as a supplement in a mineral leak block. But if we use the leak block to reduce for this, we need to careful about the right amount and dosage when using bromoform. So for this reason, we conduct to do the project about characterizing intake, leaking behavior, and performance of seed provider seed feed containing block supplement. And for the objective of the study, we will aim to analyze the seed behavior, focusing on their leaking behavior when exposed to seed feed containing block, and also including the frequency, duration, and pattern of leaking to determine in correlation with supplement consumption. And I have some request questions for this project. So first, are the current low emission leak block formulation for palatable to seed? And what is the consumption of leak block? And is there any difference between treatment and control block? And what is the behavior of seed when interacting with the leak blocks? The last question will be, does seed feed containing leak block affect health and performance of seed? So for trial information, we have the experimental, we'll have 71 merino seed and divide in mob of 35 and 36 seed. And we will conduct a Turnbridge, Tasmania, over the summer 2024 to 2025. And for material and method, we have the experimental design. So the experimental design will acclimation period followed by 2 experimental periods. 2 treatment applied in each experimental periods at a crossover design, include control block and seaweed block. And we have duration, and in each period will be 30 days. And that is a picture from the Anadel farm. And also we divide into a partnership here, and we put the location from north in the south, and have the water chart, RFID and block, and the silage feeder. And then we put the seed from 35 and 36 seed inside the partnership. So the reason why we put the RFID and block between the silage feeder and water chart, because we want to seed easy to access between the feed and the water. And also when we do the project, like in the video record, I see that when the sheep will come to drink water, and after that they will need to consume the leak block. And for the data correlation, we have intake of block and animal behavior. So behavior will record by GoPro during the period time and randomly in the morning or night of each treatment period. Leaking frequency and duration were logged. And the health status of the sheep was monitored by stock manager and researcher. And that is the real picture in the real farm when we start to do the experimental. And this one is the RFID and block and water truck in the silage feeder. And also it's the real picture in the farm. And we expected the daily Bromoform dosage. We block the weight, block weight will be 17 kilogram, and CFIT at percent of block will be 2%. CFIT will in gram will be 0.34, and we expected block intake will be 7.5 gram per heat per day. And also I have the video, I take it from farm to easy understand how the behavior of sheep when they interact with big block and how we start to record the video. How to... I'm sorry, okay.
Media player. Media player.
Oh, I put already put. Okay. Yeah. So we just put the camera in the pen in the RFID block and then record the behavior of sheep when they consume the lick block. And then for the result, I have 4 results. First one is the licking behavior, lick duration, supplement intake, and the live weight of sheep. So firstly will be the consumption behavior, is the licking behavior and the non-consumption behavior. So when I see the video record, we start to do in the farm, I see that they have the different behavior between when they consume it and when they non-consume it. So for consumption behavior, I saw that they will chewing, licking the lick block. And the most activity when they consume it will be the chewing in both control and treatment. But you can see that in period 2, the chewing is the highest number from treatment block. And also for non-consumption, I found that they will hit bug, head rub together, head bug, head rub, kick, and head bug and kick. But the most activity will be the head bug in both control and treatment block. And I have the video here to show how the behavior in sheep. Yeah. And that's why how we record the video. So they just hit rub in the iron bar, chewing, and then the head rub. It's so funny. And for average intake consumption assessment, we divide into 4 assessments, include for 2 periods. And you can, and I just analyze the, so the color will be the different level of the consumption. So the yellow color will be the lower consumption per gram per head per day. And the green one here is the will be the fit in the predict initial consumption we expect. With around 17.5, and the orange one is the highest number, but when we call the average, will be stable. And for the estimate intake, I just divide in 2 periods, and 2 periods include two blocks because we use a cross design, so we just swap control and treatment block, and then we swap over again, and then I have the data here, so you can see that is the peak initial in the period 1 block 2, and then in period 2 is just stable. And then when the average intake of CFIT block over the treatment period was 20.8 grams per heat per day, is nearly for the expected initial intake we expect. And for the lick duration, For the leak duration here, the map below is combined total time in second captured block, and we split it between the consuming and not consuming in second and percentage. So we record average at about 4-hour block period, begin with 6 a.m. to 10 a.m. And then it can be seen that the percentage of spent in consuming time is more than non-consuming, and also same with the second. And for the live weight of seed, so I have presented them as the percentage of pre-treatment and post-treatment on the knot, on the sound, in the frame size and composition, as can be seen from the box block before and after the trial. And we have the difference because we're missing the morning feed on post-trial weighing due to sharing, and large variation in frame size driving live weight spread and train over trial period. Some expected due to more dynamic in the feeding scenario with gusty and incisive, and arm farm was to reduce weight for joining as some is too fat. But otherwise, the result will see that like the seed will be healthy all the experience when we're doing this. And I just found something for the key finding which answer the question research. So first one is the low emission leaf block formulation is palatable to the seed. And second, chewing behavior is the predominant when sheep consume the leaf blocks. And average intake of seafood block is close to the target intake. And we don't have no obvious issue observed in this study. So for recommendation, I will recommend that we need to consider season, environment, and pasture when offering leaf block. And we can do like further study on long-term performance, cost benefit, and other production measures such as pool of reproduction. And also we need to apply learning to new formulation of leaf block and measure methane emission inside the digestive of the sheep when they consume the leaf block. And also, because I have a question, like when I see the video on the sheep, I saw that the behavior will be chewing, licking, but I don't know what the behavior different in cow or beef cattle. So that's why I just recommend that we can do same work in beef cattle to look at potential behavior difference. And this is my reference. And that is all about my project present. And I just want to say thank you with my supervisor team, Dr. Ape, Dr. Richard, Mr. Rohan, and also I just want to say thank you for all the staff, TIA, and also Anadel Farm, Pre-Manager, and C Forest Company. Thank you so much. And I'm ready for the question now.
Anybody else? Well, please applaud. Thank you, Thuy. Thank you for your presentation.
Yes, thank you so much.
Next, Aman, would you like to step up, please? Aman Ojha, his project is practice change to improve soil microbiological properties in apple orchards. Thank you.
Thank you. Thanks, Eric. Good afternoon, everybody. I'm Aman Ojha. Have done my research on the topic practice change to improve soil microbiological properties in apple orchard. And this project was supervised by Ramandeep Singh Sidhu and Brianna Walker. I would like a special thanks to Dr. Saint Paul for helping me throughout this project. So let's start. When we say apple production in Australia, it is like one of the major horticulture crop in Australia, having like valuation of nearly $650 million. And we look in Tasmania, it contributes around $67 million of that total share market of apple production. There are nearly like 400 commercial growers in Australia, and 2% of the total production of apple is exported to other countries, and then rest of are used and then in Australia. And then in Tasmania, the main production site are Tamar and Huon Valley region. So for my experimental design, we have chosen 2 blocks as we are doing this project in 2 cultivar, that is Jazz and Southern Bliss. The Jazz was around more than 12 years old plants, and then Southern Bliss are a bit younger with like 4 or 5 years old. And both of the blocks have 6 replicas, and in Jazz we have like 8 trees per blocks, and in Southern Bliss we have like 10 trees per block. We have taken a random block design for this project, and we are using 3 different treatment in Jazz. That is pear strip. We have given a herbicide that is organic herbicide, because all of this project is going through as organic input. We haven't used any of the non-organic substances to the production. And we have also used compost mulch and legume grass mix in Jazz variety. And same in the Southern Bliss, we have used control, that is the grower practice. Whatever the grower are doing in their other blocks, we have just followed that same practice. And compost, same as the Jazz block. And the 2 different, one is pea straw mulch and mow and throw. Mow and throw is like mowing the interrow, whatever grasses are in between the interrow, we are using that as a treatment and just putting under the line of the tree. And you can see in the picture, the left side of picture, actually my left side of the picture is compost, and right side is pea straw mulch treatment under the line of the trees. And this project is a part of PIPS, which is in its fourth year of implement, and it has like 2 or 3 more years to go. So for the project, I have taken some research questions, that is like how the soil amendment influences the soil microbial activity in the apple orchard, and does this apple cultivar, that is Southern Bliss and Jazz, responds differently with the identical treatment that we have in the both of the variety in respect to soil microbiological properties, and what is the impact of this soil amendment on the microbial indicator. We have taken a few indicators like biomass, diversity, fungal to bacterial ratio. And also we have used one of the TIA research team thing, that is Quill Enos, to detect the soil carbon emission in the field, but it also like uses different parameter, but we are focusing only on carbon dioxide emission, as we can say like the microbes, if there's more microbes, there's more carbon dioxide emission. So we are looking that in the soil amendment as well. And my project aim was to evaluate the effect of specific orchard management practice, that is like compost, legume grass mix, mow and throw, and pea straw mulch herbicide on soil microbial health. And for this, we are measuring like the microbial responses, such as microbial biomass, enzyme activity, fungal to bacterial ratio, etc. And for the methodologies, I have done this in like 3 ways. So first one, to access the microbial activity in the soil, I have done the FDA analysis, that is fluorescein diacetate. For this, the soil sample were taken from the orchard, and then it was, the environmental condition was maintained because we used the freezing condition to store the soil sample, and then I prepared some of the sodium phosphate buffer and FDA substrate, and then 1 gram of air-dried soil sample was taken, and then 50 ml of buffer and 0.5 ml of FDA substrate was used, and then the sample was incubated for 3 hours at 37 degrees centigrade, and then the reading was measured of the supernatant in the 96 well plate in a spectrometer. And the other was the microbiology lab Australia data, that we collected the sample in same way, and then we used Ziploc bags and logged these in the ski and ice pack, and then send those samples to microbiology lab Australia for the testing, and we got the data from there, and I used ANOVA to access the treatment effect on those orchard. And then other one is like we deployed the Enos in the field, and then we left the Enos for like around more than 24 hours to see the carbon dioxide emission in the and then reading was retrieved from the TIA research team, and the data was given by them, and then I used ANOVA to analyze the data as well. So coming to the results, for the FDA analysis, we saw the higher fluorescein released in herbicide. So what does this mean? Like higher fluorescein release means there's higher microbial activity in herbicide. Basically, the herbicide is like the organic herbicide, as I have already told you. And there was slightly lower microbial activity, as you see the release of fluorescein. That is, and then we see the interesting thing is like compost shows like very really low microbial activity, as it was slightly lower in fluorescein released. And so there was statistically significant difference, but not that high as we expected in ZAS block. And same in Southern Bliss. We saw like in this Southern Bliss, compost has like the highest fluorescein release, showing that there is more microbial activity going around the compost, while there's control, that is the grower practice, shows the lowest release of fluorescein, followed by pea straw. And yeah, so grower practice was having, we can say like they have like lower microbial activity going on around the control. And those p-value was like less than 0.05 that shows like this. Our result was highly statistically significant among the treatments. And this is the data I got through the Microbiology Lab Australia. And in that, we have taken around 5 parameters to compare the treatment effect. So the first one was total bacteria, and then total fungi, total microorganism, and fungi to bacterial ratio, and microbial balance. So in that case, we saw that herbicide and grass legume has higher microbial activity, while its compost again shows the lower values, especially in the like fungi and microbial balance. And only fungi to bacterial ratio was significantly affected, as I got the value of p-value of 0.003. And other parameter in the ZAS variety didn't show any significant difference because the p-value was greater than 0.05. And in Southern Bliss, we again saw the same, we again tested the same parameter, and untreated the grower practice and the pea straw showed the higher microbial activity, especially if we see that fungi, total fungi and total bacteria count, it was much higher than other parameters. And then we also saw that again, compost didn't perform well in the Southern Bliss, showing the lower values, especially in bacteria and microbial balance. As we can see in the graph that there is less variability among them. And fungi, microorganism, and microbial balance was the significantly affected in the Southern Bliss among the treatments, which shows like there is microbial activity going around the treatment. And then fungi to bacteria ratio again has no difference in Southern Bliss. That might show us like there is the microbial activity is variety specific going on around the orchard, maybe because of the root system of the cultivar. Also, the quill deployment we used in the orchard showed the high CO2 release in control treatment, because we didn't use the quill deployment in the Jazz variety. We have only used that in Southern Bliss. And then we observed like the higher CO2 release during in the controlled treatment, which is the grower practice. And straw and mow and throw was like moderate, like it was not that lower than the control, but again, the compost here shows very low carbon release. That again signifies that there's less microbial activity going around the compost. And then statically, the significant difference in CO2 releases was, these values were confirming there was significant difference in CO2 release. That again simplifies that we have different microbial activity going around the treatments. So what we can conclude through this is microbial activity, that is through the FDA hydrolysis, there was varied significantly across the treatment. There is significant effect of soil amendment on soil microbial activity. Control and straw shows like highest CO2 emission through the quail data, showing there is active microbial respiration going on in the treatment. The result also support that we can go through the use of the biological reduced input practices to enhance soil microbiological health. And also this is like the fourth year of the PIPs, but we have changed, there was like some changes in the treatment from the third to fourth year. So I can say like there might be few things that in the microbial activity that may increase in future, because it might take some time to go around the soil. Yeah, so that are the literatures I went through for my research. Thank you.
Thank you very much. Thank you. Please. I'd like to introduce you all to Harry Pergel, evaluation of nutrition-rich biochar on forestry seedling growth and soil properties. Harry, thank you so much.
Thank you.
Please.
Hello, everyone, and good afternoon. I'm Hari Paurel, and I did a project where I evaluated the application impact of nutrient-enriched biochar on forest seedling growth and soil properties. I have been supervised by Dr. B.N. Kadas and Dr. Harriet Walker. This project was funded by Sustainable Timber Tasmania, AG-SPAN, and TIA. So to say whether this project was important, I will definitely say yes. So let me explain about the background story behind conducting this project. So we all know that forestry is one of the biggest industry of Tasmania, but currently this industry is facing like two-way challenge. First one, this industry is facing how to manage the high amount of forest harvest residue. And in other hand, it is also facing the management issue of high excessive use of chemical fertilizer due to the increasing demand of wood and timber. Similarly, like the forestry industry, another biggest industry of Tasmania, which is dairy industry, is also facing the similar problem of managing its bio-waste, that is dairy effluent management. So for both this, there may be one common solution, which is a nutrient-enriched biochar. So our industry, Sustainable Timber Tasmania and AG-SPAN, comes with an idea to utilize those forest residue and the dairy effluent to make a nutrient-enriched biochar. This is get when the wood residue from the forest is subjected to pyrolysis to get a biochar, and then the dairy effluent is inoculated to make it nutrient-enriched. So this nutrient-enriched biochar is a sustainable approach that can solve the biowaste problem of two major industries, that is forest industry and dairy industry. And in other hand, if we use that nutrient-enriched biochar as a soil amendment, it is believed that it can improve the soil properties, improve the plant growth, and also help in the carbon sequestration. But there is a less known about this product, which is nutrient-enriched biochar. It is actually a novel product, and there is very less known about it. And most of the studies that has been in a biochar, it was only done in the agricultural crop, and very few was done in the forestry or in the Tasmanian condition. And also, there is insufficient data to support that this rate will be appropriate to apply in the plant. And also, there is less known about the appropriate pH range and also to its comparison with the mineral fertilizer. So we conduct this experiment with a major objective to evaluate the benefit of nutrient-enriched biochar on forest shielding growth and soil properties. And we expect that it will help us to determine the application rate of nutrient-enriched biochar for better forest shielding growth and soil quality, and also to examine the potential for nutrient-enriched biochar to replace or supplement the use for chemical fertilizer. So for this experiment, I did a pot trial using a eucalyptus seedling as a model of forest plant. And then I transplant it in a soil which is native to the Tasmanian forest. And I apply there are 5 different types of nutrient treatment. In treatment 1, I use a control where there is only native soil, not any supplement of fertilizer and nutrient-enriched biochar. In treatment 2, I use 20 gram nutrient-enriched biochar. In treatment 3, I use 40 gram nutrient-enriched biochar. In treatment 4, I use 20 gram nutrient-enriched biochar plus 10 gram chemical fertilizer. And in treatment number 5, which was the last one, I use only 20 gram fertilizer. This 20 gram fertilizer is the standard rate of fertilizer where the industry is using while planting the forest plant. And this fertilizer is, I use multigot as this fertilizer, which have a nitrogen, phosphorus, and potassium ratio of 17:7:14. I did experiment in a randomized complete block design, which was oriented from south to north, and there are 6 replications. And then automated irrigation system was provided. And this trial gone for a long month. And then to know about the seedling response, I measure the seedling growth parameter where I measure the growth rate and the stem diameter and the whole biomass of the seedling after harvesting that one. And then we are also measure the soil chemical properties, that after application of that nutrient, how it changed the soil chemical properties there. And then after measuring the data, I use 1 factorial analysis of ANOVA in R Studio version 4.3.2. So talking about the result, in growth rate, we found that there is no any significant difference among the treatment except between the day number 15 to 30, where we get that the growth rate of seedling where application of nutrient-enriched biochar, 20 gram nutrient-enriched biochar plus 10 gram fertilizer was shown high. Otherwise, in all other treatment, the difference was not significant. So the response of treatment in stem diameter, we found that the stem diameter was significantly different among different treatment after day number 63. And it was found high in the seedling where the application of 20 gram fertilizer was applied, but the response of seedling where we applied 20 gram nutrient-enriched biochar plus 10 gram fertilizer also significantly similar with that one where we have applied 20 gram fertilizer. But in control and 20 gram nutrient-enriched biochar only and 40 gram nutrient-enriched biochar only, the difference, there was no any difference, significant difference. So the response of applying treatment in leaf chlorophyll content, we measure this leaf chlorophyll content to check the seedling vitality. In this also, we didn't found any significant difference across the different treatment after 4 long period, but we found the significant difference only at day number 21 and 63. In day number 21, the seedling which were 20 gram and nutrient-enriched biochar plus 10 gram fertilizer was applied, it was shown high, but in day number 63, the seedling where 20 gram fertilizer was applied, the leaf chlorophyll content was high there. If we talk about the 20 gram nutrient biochar only, 40 gram nutrient biochar only, and control, the graph show that the seedling where we have applied 20 gram nutrient biochar contain more chlorophyll content throughout the period, but it was not significantly different than other ones. So in seedling biomass, we found there is a significant difference between treatment. In stream biomass, either it was fresh or dry, we found that the seedling which was treated with 20 gram fertilizer showed a higher mass, which was also significantly similar to the seedling stream biomass, where we have applied 20 gram nutrient biochar plus 10 gram fertilizer. Whereas in control and 20 gram nutrient biochar plus 40 gram nutrient biochar, the response was similar. So to summarize the result, we found that there is no significant difference in growth rate and chlorophyll content across all the treatment after 4 months of growth. And there was a significant increase in stem diameter and biomass observed in seedling treated with standard rate, that is 20 gram chemical fertilizer, and in the seedling where we have applied half rate of fertilizer, that is 10 gram and 20 gram NAP. But we didn't find any negative impact of using nutrient-enriched biochar. So to conclude, we can say that application of nutrient-enriched biochar has no any positive or negative impact on seedling growth when applied solely. And we are still waiting on a soil chemistry result, and the reason why the nutrient-enriched biochar is not effective could be due to the negative chemical interaction, maybe like pH or other factor. And this trial was limited to 4 months. So the benefit of solo risk nutrient-enriched biochar may be observed later, because the biochar is actually release the nutrients slowly over a period of time. And our industry, who is funding this project, that is Sustainable Timber Tasmania, they also doing the field trial, applying the same treatment, and there we can find the benefit of nutrient-enriched biochar after a few years. So after conducting this research, I like to recommend future students to do a glass out trial using greater number of application, and also using different bio-waste stream like CRCin plus biochar and more concentrated refuse plus biochar. And for industry, I like to recommend a long-term evaluation of nutrient-enriched biochar from different waste stream on forest seedling growth in the field condition. So lastly, I would like to acknowledge my supervisory committee, Dr. B.N. Kadas and Dr. Harriet Walker, for their continuous support and valuable feedback throughout my project. I also like to thank Dr. Mary and Dr. Thomas Baker from Sustainable Timber Tasmania. I also like to thank Miss Karen Enkiller from AG Spain. And I also like to thank Dr. Caroline K for managing all the necessary material while measuring the data. And I also like to thank my friend Prajita Gimira and Shilpa Koirala, who are helping me during data measurements. Thank you so much, everyone.
Who's our last speaker? And she's going to talk to you about, leveraging weather data and site-specific information for adaptive spraying in vineyards. Take it away.
Hello, everyone. My name is Chu Zhang. I'm going to present accessing weather-based models for efficient spraying management in vineyards today. First, I would like to say a big thanks to my supervisor, Dr. Shahin, and all the staffs who give me helps from TIA and my family and friends. Okay, let's start. This is the order I'm going to present. The grape vines are cultivated extensively worldwide, with wine-producing regions spanning across continents. Fungal disease such as powdery mildew, downy mildew, and botrytis branch root are among the primary causes of reduced grape yields and compromised fruit quality. To protect vines, growers often implement intensive chemical application strategies, such as weekly or fortnightly spraying. While this approach is generally effective in disease control, it incurs significant economic costs and poses critical challenges like pathogens develop in resistance, while the environment and worker health face potential risks. Notably, grape cultivation stands out in agriculture for its heavy reliance on fungicides, with vineyards applying exceptionally large quantities of chemicals to combat mold-related diseases. Consequently, there is a growing global emphasis on sustainable grape disease management, maintaining yields while reducing chemical inputs. This necessitates improved control strategies, such as employing more scientific diseases prediction models and optimizing spray timing to ensure precise chemical applications at appropriate times and locations, thereby minimizing unnecessary treatments. And now we are going to the common grape disease and their impacts. There are three major fungal diseases in vineyards, and first is powdery mildew, as the most widespread grapevine diseases globally. It's thriving under warm conditions with high humidity. It forms wide powdery patches on affected leaves and fruit surface. This severely inhibits photosynthesis, stunts berry development, and compromises wine quality. Like if untreated infections, it will lead to significant yield losses. The second one is the downy mildew. Unlike the powdery mildew, this pathogen requires water droplets or prolonged leaf wetness to infect. The industry often uses the 10-10-24 rule to assess initial infection risks. 10 millimeter of rainfall, temperatures above 10 degree, and 24 hours of stained moisture. Infected leaves develop yellow oil stain spots on the upper surface, and poor control results in severe early leaf drop, berry shriveling, and notable yield reduction. The third one is botrytis. It prefers cool and humid environments. It causes grape clusters with gray mold, causing like berry root and browning, directly reducing yields. Even minimal infected grapes in winemaking can introduce flaws like oxidized or moldy off flavors. In cooler regions like Tasmania, this is a persistent annual challenge. These three diseases each flavor distinct environmental conditions. Effective measurement requires understand their mechanisms, enabling early prediction and targeted prevention and control measures. Then I'm going to talk about the evolution and mechanism of disease models. Grape disease prediction models have evolved continuously with technical achievements. Early growers relied on empirical rules like damp and rainy weather promotes malt growth to implement broad spectrum control measures such as routine spraying during rainy seasons. By the mid-20th century, the first generation index models emerged, exemplified by the Googler-Thomas model, which dynamically quantifies risk by accumulating points hourly when temperatures range between 10 to 27 degrees with leaf wetness duration exceeding 6 hours, while resetting the index upon exposure to high temperatures or dry conditions. The new the New Zealand's Bacchus index optimized for botrytis not only tracks a cluster wetness duration, but also pioneered a dryness penalty mechanism, deducting 15% of accumulated risk after 2 consecutive dry hours to precisely simulate the interruption of fungal infection. Concurrently, degree-day models which predict infection windows by accumulating effective temperatures gain the traction for their simplicity, but it required localized celebration of temperature with the rise with the rise of artificial intelligence and big data, Bayesian network models emerged, linking multiple meteorological factors to disease or accuracy through probabilistic frameworks. Trained on historical multi-year data, these networks learn how variable combinations in influence powdery middle outbreak probabilities, outputting a dynamic risk estimates rather than rapid index values. This probabilistic approach aids decision-making by quantifying both risk levels and the prediction confidence. Additionally, Bayesian models continuously self-update, refine accuracy with each season's new data by integrating diverse factors and adapting to new information. Bayesian networks demonstrated superior adaptability in regions with complex microclimates like Tasmania. Next, we are going to talk about the comparative of the models. In these slides, we compare the characteristics of several models using this table. First, consider the GTI is advantageous lies in its simplicity and practicality. It requires only temperature and leaf wetness data for calculation, making it highly accessible. However, since its shepherd were established under California conditions, its accuracy declines when applied directly to the cooler, humid regions like Tasmania, necessitating parameter adjustments. Next, the Bacchus index is the model especially for the botrytis. Its strength includes a dryness penalty mechanism. If the weather turns dry during the monitoring period, previously accumulated risk is partially reduced. However, it's exclusive actual load for botrytis management and requires significantly higher equipment standards for data collection. Finally, the Bayesian network model it's accessed by integrating as many relevant factors as possible, like temperature and humidity, rainfall, leaf wetness, cultivar resistance, and sunshine reflection. It makes it far more adaptable in complex microclimates like Tasmania's diverse valleys, and slopes and localized weather variations. Its accuracy significantly surpasses traditional index models under such conditions. Another major advantage is its ability to continuously learn and improve over time. However, the Bayesian model's drawbacks are evident. Like constructing such a model demands extensive high-quality historical data and expert knowledge. Most growers cannot develop a Bayesian network independently, and typically require support from research institutions or technically companies. Additionally, its internal reasoning process is unclear to users, making it difficult for farmers to understand why the model produces specific results. Barrier it actually is a barrier to adoption. Furthermore, operating a Bayesian model typically relies on computer systems and specialized software. Real-time field data measurement and probability calculations require substantial equipment and financial investments, which may be impractical for many growers. So my aim is actually to find out to what extent the farmers accept the models and how I can let them to accept them. From my interview to the farm, while the old method is very common. For instance, spraying every 10 days for fortnightly offers convenience, but risks mistimed applications, spraying too early or spraying too late. And conversely, some growers adopt a reactive spraying strategy, treating only when disease symptoms appear. It's too late to prevent initial infections and losses. The second approach relies on models or decision support systems. I studied this from the European articles. So some of them are using the DSS systems, but not so common. And so this is the challenges in promoting model adoption. And I try to run the GTI in Excel. This is the results I got. The model is based on the GTI principle, but localized for Tasmania's cool climate from 2024. While experimental validation was not feasible due to resources constraints, dynamic risk assessment was achieved through simulation. When leaves remain wet for over 6 hours with temperatures between 10 degree and 27 degree, conditions favorable for pathogen activity, the model increments is a risk score hourly. If afternoon temperatures exceed 35 degree or humidity drops up roughly, the model interrupts or sets the risk score, simulating inhabited pathogen reproduction. This creates a dynamic risk curve reflecting weather-driven disease. The risk index is divided. Let me pass this. Like you can see from the picture, the x axis represents time, and the y-axis shows the risk index. Initially dry weather keeps the curve low, the green zone. Consecutive that wet days drive index accumulation, pushing the curve through the yellow median risk zone. Sorry. Like these pictures can show the high risks and the low risks when we run the models. And in the future, integration of additional variables through trails could evidence these weather-driven precision management systems in cool climate regions like Tasmania. Okay, overall in Tasmania vineyards, the advanced Bayesian prediction models have demonstrated superior performance over the simple degree day models in forecasting the grape disease outbreaks. Through the limited data availability remains a key challenge. Nevertheless, the weather-driven forecasting tools have already proven their value by enabling farmers to make proactive disease management decisions. To advance model adoption, several steps are essential. First, improving the user friendliness of model tools, such as developing intuitive mobile app interfaces with clear risk indices and actionable recommendations. Secondly, localizing existing models like GTI and Bacchus by recalibrating their parameters for Tasmania or specific sub-regions to align predictions with local realities, thereby building growers' trust. Third, enhancing training and demonstration programs through side-by-side trail plots showing reduced spray applications without compromising this control to convince growers with tangible data and outcomes as younger generations of growers become more technical and the sensor costs decline. The precision spraying via decision support systems will likely gain attraction, benefiting both farmers and environmental sustainability goals. Ultimately, in integrating advanced disease prediction with precision agricultural techniques and other sustainable practices, such as improve the canopy management and smart crop sequencing or rotation, cultivation of disease-resistant grape varieties, and diversified planting can drastically reduce the reliance on the fungicides while enhancing long-term resistance to disease pressures, thereby supporting more sustainable viticulture in the precision agriculture area. Thank you.